everybody. We've had a great lunch here with a lot of discussions, talking, looking at exhibitions, and we've also all said that what a fantastic meeting, what a lot of nice new things we learned about the history of NIDCAP in Sweden, but also about epigenetics. And we are very lucky because we will hear more about epigenetics this afternoon. And we will also be part of the very glorious, or say, inauguration of the new NIDCAP Center later this afternoon. Is there any comment from the morning? You have all been able to hear and see. I think we thank the technicians. Everything has worked very well. So with that, I would like to introduce the next speaker, Professor Rosario Montirosso, who is a developmental psychologist and a researcher in Bosisio Parini in Lombardy in Italy. And Professor Montirosso will speak about epigenetic protection. I know the previous speaker said that this is of course of a great interest to do this, and the role of caregiving touch and body-to-body contact in the early interaction. Please welcome Professor Montirosso. We are happy that you are here, and we can now see you very well. Can you hear? Oh, we can hear you too.
Thank you.
Yeah,
Thank
please.
you. Okay.
Yeah.
Can you hear me?
Yes, we can hear you.
Okay.
Yeah, and see you.
Okay. Hello, everyone. It's a big pleasure to be here, even if only in a virtual modality. But okay. First of all, let me thank all of you for attending the meeting, and also I would like to thank Stina and Agnes for inviting me here today. Before I go on, I would like to say just a few words about this bad times. Maybe most of you know this picture. It's a part of a video of a Ukrainian NICU newborn and a healthcare professional relocated to a bomb shelter. The shelter is on a lower level of the hospital buildings in Dnipro, a city that was the target of Russian strikes. The video showed nurses who soothe some babies, and a few babies are not able to breathe in without assistance. Lacking life support machine, staff kept those babies alive using inflatable bags to deliver breath after breath after breath. Nearly all of us cared for sick babies. We spend hours in our hospital tending to these babies. Our patients are babies born premature, babies with birth defects, babies who struggle to transition to life outside the womb, babies who cannot breathe without support. Those babies' lives depend on those healthcare professionals continuing to work assisting their patients' breaths in that shelter as the war ravaged the street above them. Maybe those babies will live, but only to live in a war-torn country, in a war-torn world. Unfortunately, there is not only the Russia-Ukraine war. And if we take a look at the data provided by the Armed Conflict Location and the Event Data Project, we see that a large part of globe is affected by armed conflict. The Armed Conflict Location and the Event Data Project is a non-governmental organization specializing in conflict data, data collection, analysis, and crisis mapping. In 2022, this organization has recorded more than 50 wars around the world, and children are impacted the most. Save of Children organization says that 1 in 6 children in the world lives in the conflict zones. More than 10,000 are killed or mutilated by bombing in war areas. 100,000 newborns died every year for direct and indirect causes of conflicts, such as disease and malnutrition. Children living in war affected countries living in constant fear experience significant violation of their childhoods. So, while the world goes on with war and the protests and the streets are full with sorrow, countless children grow up in the normality of war, now nothing but suffering of their stolen childhood. As a member of healthcare professional community and of a scientific community, I feel that we should claim clearly that the war is not a solution for solve conflicts. And for sure, the war is not a solution to grow up kids in freedom without hate in their hearts. Uh, okay, now I go on with my presentation, which is about epigenetics protection. It's just to be strange to speak about protection uh, while we live the war. Anyway, we have heard Agnes and Livio talks about epigenetics, really good talks. But what means epigenetics protection? Generally speaking, it's just a way to discuss epigenetics from a different point of view. And as you know, most behavioral epigenetic studies have focused on the biochemical markers of early diverse experience. However, if DNA methylation is responsive to environmental cue, then it's possible that even positive experience can have an impact on epigenome. In this view, epigenetics processes are considered as a biological mechanism of resilience, linking developmental experience, such as parenting behavior that supports infant development or buffer potential detrimental effect of early negative experiences, to later life behavioral outcomes. Epigenetic protection should be considered part of a dynamic process related to developmental plasticity. Indeed, the developmental plasticity represents an adaptive process by which a specific genotype can give rise to different phenotypes according to the environmental experience they live. To put it in another way, developmental plasticity is linked to resilience. On the negative side, once the impacts of environmental experience overcome the capacity of resilience, impaired or disrupted developmental outcomes will be result of incomplete buffering. On the positive side, resilience serves as a process through which organisms achieve a positive adaptation to environmental changes and prepare for future adverse experience. So epigenetics change in the brain, in brain structure and function might contribute to the developmental plasticity, but in terms of adverse developmental outcomes or promoting resilience. This brings me to the next point, that is, epigenetic process represent important mechanism underlying developmental plasticity in response to environmental exposure, which can use the epigenetic change reflecting two aspects of that plasticity. Toxicity effect, as well as adaptation in the process of development. Epigenetic toxicity is about exposure-induced epigenetic modification leading to undesirable health effects on organisms, potentially underpinning the predisposition to disease due to environmental exposure. Exposure-induced epigenetic toxic modification appear to result from insufficient buffering of perturbation. In other words, the toxicity of environmental factor exceed the capacity of resilience, overcome the capacity of resilience. However, the effect of early experience might be much more complex, given that in the most cases, babies, infants, are exposed to both negative and positive experiences. Thus, we should consider the role of developmental-oriented experience as well. In other words, epigenetic change caused by environmental exposure maybe not always lead to impairments, but may also the individual to achieve positive developmental outcomes through appropriate adaptation. Here, the next point. Oh, sorry. Before I elaborate the view that the epigenetic process may serve as a mechanism for resilience, I would like to show how epigenetic toxicity might constitute a persistent biological memory of early experience, which could lead to impairment in developmental plasticity. Let me show briefly what happened in prematurity when environmental experience overcome the early capacity of resilience of an infant. Preterm infant, Livia showed us, preterm infant experience several NICU-related stressors such as procedural intervention, reduced social contact, and so on. These NICU-related stressors have been found to result in birth to discharge, increased serotonin transport gene methylation, which in turn is associated with suboptimal outcome in the first month of life. Not only we have found that epigenetic change go behind the first year of life, and they affect behavioral outcomes in the preschool age, for example. In our longitudinal study, we assessed the emotional regulation during an observational procedure designed to assess children's emotional regulation at 4 years of age, which include 4 stress episodes. And as you can see in the slide, preterm show greater anger compared with full term. You see here, full term control in response to this stress episodes. And interesting things was that emotional dysregulation in preterm preschoolers was predicted by methylation at the NICU discharge. Thus, NICU adverse experience, which possibly overcome the resilience of these children, have an impact on the epigenetic process related with adaptation to a stressful situation, which was observed several years later. However, at this point, one can wonder if the epigenetics change at the NICU discharge are only predictive of long-term outcomes, or if they remain stable during the development. To put it in another way, one could think that emotional dysregulation at 4 years is also associated with epigenetic change at NICU discharge. That is, early epigenetics change are a mechanism underlying the programming of the stress response. But a good question could be, do early epigenetic change remain stable across the development? Do the epigenetic markers associated with early adverse experience in preterm infant constitute a permanent biological status? This brings me to another question. Does epigenetic status at 4 years linked to emotional dysregulation observed in these children? In practice, this would mean that emotional dysregulation observed during preschool age could be also modulated by the epigenetic status at 4 years, and not only by epigenetic change observed at the NICU discharge. So, as you can see in the slide, the emotional dysregulation at 4 years was also associated with a DNA methylation at 4 years. This might suggest that in some children, DNA methylation is stable from NICU discharge to preschool age, and maybe even in a later period of life. In other words, it seems that epigenetic toxicity might contribute a persistent biological memory of early experience, which could lead to impairments in developmental plasticity. But fortunately, you know, development is a very complex process in which several factors contribute to favor the adaptation of the organisms. A growing body of study claimed that positive effect of environmental exposure through epigenetic mechanisms. Animal studies highlight that parental environmental exposure can benefit the offsprings to achieve permanent epigenomic adaptation. For example, breastfeeding represents an important factor during which the DNA methylation can be sensitive of positive change with long-term beneficial effects. Therefore, in the epigenetic study is needed to consider the role of a protective factor and how long their effect could be stable. Of course, there could be many factors might be associated with epigenetic protection, but as you know very well, in the early years of life, the caregiving touch and the body-to-body contact could play a special role. The sense of touch is the first manner of contact with the external world, providing a foundation for the developmental of sensory motor skills and social affective behaviors. It represents a link between the body, internal physiological states, and the external world, sensory stimulation, suggesting it may be critically involved in the developmental plasticity. Thus, in this part of my presentation, I would like to talk about the role of caregiving touch as a epigenetic protective factor with a significant impact on infant neurodevelopment. I will review studies that address early caregiving touch in association with DNA methylation, posting that the mother-infant touch acts as epigenetic protection mechanism. Before I move on, I would like to address briefly this topic from a general point of view. Therefore, I will not focus on the benefit of touch and the skin-to-skin contact in preterm infant, because I'm sure you are very familiar with this finding, with this research. Rather, I want to provide the recent findings useful to discuss in more depth the implication of a caregiving touch in the developmental plasticity. Of course, one can wonder why the caregiving touch the body to body content it's so important. It's a very stupid question, I know. The response is very simple. And the first months of life, early mother infant exchange are primarily based on the embodied interaction, during which mother use tactile stimulation and the body to body contact to support infant behavioral regulation, and such content also encourage the infant to form an emotional bond with caregivers. And interesting thing is that neurophysiological studies have documented the existence of a dual touch system consistent of two parallel neural pathways, one for discriminative touch and the other one for affective touch. Affective touch activities active a population of fiber called C-tactile afferents, which respond in optimal way to be the low force and the velocity typical of nurturing care. These C-tactile fibers convey effective and socially relevant information to the brain. Along with other components of parenting, caregiving touch can calm infants and mother, promoting the sensation of pleasure and positive affect. Uh, Importantly, sensation from the skin as the earliest developing sensory modality. The somatosensory system is the first to develop from maybe about 8 weeks gestational age, and several brain area processes tactile information. As you can see in the slide, discriminative tactile information is processed by the primary somatosensory cortex, while affective touch is processed in the insular cortex. Not only uh, other areas, brain region are involved in affective touch. For example, the superior temporal sulcus, anterior cingulate cortex, and all amygdala, prefrontal cortex, but all this region are involved in tactile information processing with reference to emotional and the social context. Uh, of course, I have no time, there is not enough time to discuss in detail the beneficial effect of touch for infant development. However, let me show you briefly in how many domains early touch for a caregiver provides with benefit for infant psychosocial and neurophysiological development. As you can see in the slide, affective touch support the development of infant somatosensory, stress regulation, and the immune systems. Affective touch also plays a foundational role in the establishment of social affiliative bonds and early psychosocial behavior. This touch related bonding effect are known to be mediated by the oxytocin system, but touch also activates dopamine and endogenous opioid systems, which support the development of social cognitive processes such as social learning and reward processing. Finally, affective touch plays a crucial role to establishing and maintain parent-infant interactional synchrony at behavioral and neural levels. And I would like to look at this point a bit, this point in a bit more detail. Uh, for example, I want to depend if the newborn physiological sensitivity to different types of tactile stimulation. In fact, one can wonder if infant perceive the difference between being held and being hugged. Yoshida and colleagues have examined this question. They recorded an electrocardiogram during a certain sequence of a hug hold session and examined RR interval, that is an indicator of a parasympathetic regulation in newborn in infants. The mother were instructed to perform 3 behavioral tasks for 20 seconds each: to hold the infant lightly as usual, hold; to hug the infant while thinking that the infant was adorable, hug; and to hold the infant very tightly, as if the mother could run fast while holding the infant. As you can see in the upper graph, the RR interval, sorry, okay, was greater when the infant were hugged than when they were hugged thickly. Infant response to a parental hug is thought to involve many brain regions, the same that I said before. So this connection between hug and brain activity maybe can support parasympathetic activity, which led to an autonomic regulation. I would like also to say that it's important to highlight that even caregivers felt positive emotion and increased the parasympathetic activity when they hugged their infant. Here we have another example about the role of affective touch in autonomic regulation. In this study, research studied the underlying mechanism of touch in terms of parasympathetic regulation measured by infant respiratory sinus arrhythmia and RR interval. They compared the 3 minutes touch period, stroking or non-stroking touch, with a baseline before pre-touch and after post-touch in diets with infant aged for 16 weeks. As you can see, compared to a non-stroking touch during and after maternal stroking, the parasympathetic regulation of infant increased while mothers stroked their infants. Thus, again, Niels Bohr showed cardiac sensitivity to different type of touch, suggesting that early touch stimulation represents scaffolding for development of autonomic self-regulation, with important implication on infants' ability to adaptively respond to the surrounding social and the physical environment. But the role of affective touch goes beyond of this aspect, supporting caregiver infant neural attunement. In this fantastic paper, researchers assessed whether touch and the proximity in caregiver infants' diet were related to neural synchrony. They simultaneously measured brain activity using functional near-infrared spectroscopy of 4-6 months old infants and their mothers in distal and the proximal joint watching condition, as well as in an interactive face-to-face condition. Neurosynchrony was higher during the proximal than during the distal joint watching condition, and even higher during, you can see here, sorry, here, okay. I was saying that the maternal condition, the maternal affective touch during the face-to-face interaction era was positively related to synchrony. So this means that touch mediates mutual attunement, synchronization of brain activities in caregiver infant diets during naturalistic interaction. Uh, what conclusion can we draw from this? You know, this evidence suggests that mother-infant synchronize their bodies, okay, during early embodied interaction, which is in turn could have important implication not only for infant self-regulation, but also a crucial. It could be crucial for bodily self-perception. Indeed, touch and body-to-body contact enhance the early subject feeling of body, supporting multisensory integration and facilitating the interoceptive sensitivity in infant. Thus, the bodily attunement can contribute to regulate the infant body states, for example, decrease heart rate, which in turn might promote interoceptive sensitivity and therefore the infant and at the hand, the infant bodily self-perception. At this point, the question is, could affective touch have an impact on epigenome? In other words, does caregiving touch and the body-to-body produce contact produce methylation pattern that place infant on a positive developmental trajectory? Uh, the study of epigenetic mechanism related to early positive event is only just beginning. But a few studies have investigated the impact of maternal touch on DNA methylation in the human in either early typical development or in at-risk condition. And let's move. Let's now move on to these findings. Breastfeeding could be considered a proxy of physical contact, and Lester and colleagues looked at more than 40 full-term healthy infants and their mothers, one half of whom breastfed for the first 5 months, and one half of whom did not. They examined the methylation of a glucocorticoid receptor gene and the post-stress cortisol concentration in infant saliva after the still phase procedure. You have seen, we saw the still procedure during the Livio talks. Let's have a look at figure. The red bars represent infants who experience more breastfeeding during the first 5 months of life. As you can see, in several places, they show the lower level of methylation compared to the infant who experience less breastfeeding. And importantly, high level of methylation was associated with increased cortisol reactivity during stress, during the still phase. So this find suggests that the physical proximity reducing is associated with the reduced stress level, which probably was moderated by protective epigenetic variation. Here, I would like to show another example. This is the only longitudinal study that I know on this topic. At 5 months, maternal engagement, included interpersonal touch, was assessed during a free play interaction. Oxytocin receptor gene methylation was assessed at 5 months and at 18 months. And again, in the slide on the right plot, we say that the quality of engagement predicted a methylation reduction in their infant more than 1 year later. So this study has important implication because it suggests that Human oxytocin system is epigenetically sensitive to maternal behavior. Uh, Specifically, higher maternal engagement may have the potential to upregulate the oxytocin system in the human offspring, which is a crucial regulator of social behavior. Maternal touch could play a protective role even in at-risk condition as maternal depression. As you know, perinatal depression has been shown to negatively impact early mother-infant interaction. However, this with the infant in comparison with no depressed mother, if it was found that stroking and rubbing could increase infant positive affect and tension also in presence of depressive symptomatology. Interestingly, maternal touch appear mirrored at the epigenetic level. Murgatroyd and their colleagues have used self-reports to examine the association between the amount of maternal stroking and the pre- and postnatal depression assessed when the infant were 5 and 9 weeks old. Higher, higher maternal stroking at 5 weeks buffered DNA methylation in infants that were exposed to the combination of low prenatal and high postnatal maternal depression. So this seems to suggest that even in the presence of the adverse condition, such maternal depression, caregiving touch could trigger a sort of epigenetic protection through a biological embedding of experience at any molecular level. And of course, at this point, one could wonder, could maternal touch be associated with the epigenetic mechanism, epigenetic protective variation in preterm infant? Uh, we have examined this hypothesis in a very recent study. The study was aimed at exploring the potential buffering role of maternal touch in moderating the relationship between serotonin transport gene methylation and infant negative emotionality during the still phase, so during the social stress. We have assessed the maternal touch, which was coded as mother ability to touch infant in gentle affection manner, included static touch. And here we can see the main findings. Maternal touch was effective in moderating the association between methylation and negative emotionality. Specifically, DNA methylation in preterm infant exposed to less than optimal maternal touch was associated with increased negative emotionality during the social stress. So again, this finding seems to suggest that when we consider epigenetic variation, maternal touch plays a specific role even in preterm infant emotion regulation. So how the epigenetic protection could work, even if it is difficult to separate tactile stimulation from other potential maternal factors, such as maternal responsiveness or maternal sensitivity. Tactile stimulation seems to antagonize epigenetic effect on the stress-related gene expression in young infant. So the early embodied interaction provides a channel by means of which the molecular biological of epigenetics can be contextualized with respect both positive and adverse infant experience. One might speculate that caregiving touch, proximity, body-to-body contact produce lasting changes in the function of a biological system underlying developmental trajectory by gene expression. This view is quite aligned with the affective touch hypothesis, which claims that nurturing touch mediated by C-active fibers play a crucial protective role in the developmental plasticity. If so, then one could wonder if epigenetics protection could have a clinical implication in prematurity. And the recent study provides us some evidence in this direction. Let me give you a couple of examples. This is the same study that Olivia showed before. This is a very interesting study. So I go fast, but the main finding says that high level of gene expression were associated with greater hypothalamic pituitary adrenal axis activity. So mother child skin to skin immediately after birth significantly reduced gene expression assessed at the discharge. And taken together, this study indicates that early mother-child contact modulate epigenetics mechanism associated with the stress response. Another interesting study has examined the methylation change of the long-intercepted nuclear elements, so-called L1. L1 are a class of DNA elements which represent a significant portion of human genomes. Usually, L1 elements are repressed in most somatic tissue to prevent DNA damage and ensure genomic integrity. In other words, L1 elements are methylated in normal human tissues. These DNA elements contribute to genomic somatic mosaicism of the brain and are deregulated in several neurological disorders that often occur in individuals born preterm. Researchers found that L1-TN methylation was associated with an early developmental care program based on enhanced maternal care in NICU. Importantly, as you can see on the right side of the slide, L1 methylation increased proportionally to maternal care received through early intervention, which was quantified as the average number of messages of infant touch, maternal touch, that infant received per week. So now I'm approaching to the end of my presentation, so let me just run over some key points. The epigenome provides a molecular explanation for the existence of a biological memory coined by early positive and negative social experience, inducing a lifelong impact. However, the epigenetic don't explain everything, and the epigenetic research raises more questions than it answers. And for example, a critical point is that it is tempting to assume that specific early life experience induce a direct causality in epigenetics change. We have long known that environmental leaves its markers on our body. So epigenetic is not a new finding. I mean, what is new is an understanding of some mechanism that translate experience into cell-specific damage. So this implies to be caution both at biological and the psychological level. First, difference in maternal care could affect the expression of hundreds of genes, suggesting a broader response of DNA to early life experience. Second, it's important to avoid any misleading conclusion which could convey the idea that there is a direct impact of early experience on the short and long-term children outcomes, without taking into account the importance of a broader social and the healthcare environment. Epigenetic has suggested that epigenetics should be considered like one of the levels to analyze the complexity of the mother-infant interaction. And we should consider the multifactorial and multi-temporal trajectory which are necessary to capture dynamism of human life. At the same time, paradoxically, epigenetic find offer us a way to avoid any reductionism. Epigenetic forces us to reflect on what it means to be by embodied interaction. Uh, as I said that before, there is an emphasis on adverse events or trauma in epigenetic research. However, findings about caregivers' touch suggest that a lot of epigenetics change are related to quotidian, daily embodied interaction rather than extreme events. Uh, the lesson we are learned from studying epigenetic protection is that a lot of epigenetic change are embodied in the mother-infant interaction. Indeed, as Chanuka said, before an infant meets mother minds, the infant meets mother's body. And to some extent, the contrary is equally true. Before mother meets infant's mind, mother meets infant's body. So I have started my presentation reminded to all of us how many children are suffering just in this moment for the lack of adult responsibility. As adults, we have a huge responsibility towards children. Protect them. Epigenetics provide us new evidence about how much to protect children is important. Maria Montessori claimed that in early childhood, the physical life of a child needs to be protected and to be surrounded by an environment that could be compared with the wrappings placed by the nurturer about the physical embryo. So rather than looking at the infant development as a result of a natural and nurture contribution or genetic and environmental causes, epigenetics in the embodied interaction highlights the open-handed nature of ontogeny, the multiplicity, the multidirectional of biological information, which together, behavior, physiological, and social research, act through the bodies and they contribute to co-create meanings in the dyadic exchange. In short, bodily encounter create interactive meanings. And for this reason, I wish you any good touch for your life. Thank you. Thank you so much.
Thank you very much, Professor Montirosso. That was for this fascinating lecture. And I just waved the flowers to you from us all. And I guess you have beautiful flowers in Italy too. It's fascinating and difficult topic, this, because there seems to be so many things interacting all the time. And did you have a question from internet? Yes. We have a digital question, so please, we take that first. Yeah.
So to Rosario, question one, not really a question indeed, but a big thank, Ross. Brilliant talk, fantastic. First, thanks for reminding us our culture role as a clinician and scientist in these crazy times. Here comes the question.
Thank you.
Many types of touch can be observed in parenting, gentle, static, playful, massage, etc. What is the most important, and how would you study this in the future?
It's a good question. Yes, there are a lot of different kind of touch. As I said before, the research said us that we have a specialized fiber for stroking. So I think that each touch is important, each kind of touch is important. But of course, affective touch is very, very important. Most of in the early developmental in the early period of the life, it's very important. But I would like to say also something about touch in premature baby, because you know very well that these babies are very immature, and sometimes dynamic touch, can be perceived by this infant like something disturbing or painful as well. So in NICU, for example, it's important to use maybe static touch rather than dynamic touch. As you have seen before, the baby can distinguish by different kind of touch, and the baby can also distinguish who provide him the touch. So infants, it's a fantastic, the body infants is a fantastic machine for receive touch. So any kind of touch is good, but effective touch is the most important.
Question from Björn Westrup.
Hello, Rosario. This is Björn Westrup. Thank you very much for, of course, very interesting. And I'm very happy that you just comment this last thing about premature babies, because that was actually the question I was intended to ask. Because doesn't this have to be individualized? Because the studies that show that strong hugs were more detrimental than softer hugs. That's interesting. But I wonder if that applies to a strong man or a weak woman, or I mean, it could be different, couldn't it? Also, you mentioned now and commented already that stroking and non-stroking touch. You said that stroking touch might be more beneficial, but for whom? Which individuals? And the same with massage. So I think actually, please comment on skin to skin, because I think if we start with skin to skin in a gentle way, and if we observe the behavior of the infant, then we can adjust the way of touching, don't you think?
Yes, I know, you know that skin to skin is very important for baby, but I would like to speak about from another position, from the mother position, for the mother perspective. How is the perception of the mother during the skin to skin? Because I think that it's very important to study, to depend the mother perception during skin to skin. We know very well that the skin to skin is beneficial for infants, but what about the mother? How mother perceive infants during skin to skin? And I think that we need to support maternal embodied sensitivity. I need to help mother to recognize their body signals, because this is the first step to be empathetic with their infant. So skin to skin is great for infants, but it's important to work and support mother during skin to skin, and help the mother to recognize what her body says herself. So sorry for my English, but the idea is we needed to work to thinking that the embodied interaction is a system, and we needed to work to infant, and we need to work in terms of support, I mean, with the mother body.
Okay, thank you very much. Is there any other comments or questions around? No, I cannot see anyone. So thank you again very, very much for an excellent lecture. And with that, we continue.